This series is for those who stay curious anyway. Welcome to the Holoverse.
Every cell in your body is holding a conversation that started before recorded history. The code running inside you right now has outlasted empires, survived extinctions, and crossed oceans in bodies that didn't know they were carrying history. We've been looking for the universe's oldest record, but we've been carrying it the whole time. Before there were libraries, there was DNA. Inside every cell of your body sits a molecule carrying approximately 3 billion base pairs of information, which is the equivalent of roughly around 200 volumes of text, replicated in every single cell you have. If you unraveled the DNA from just one human cell and stretched it from end to end, it would measure about 6 feet long, and you have approximately 37 trillion cells. The library isn't stored in just one place. It's distributed everywhere simultaneously. What makes DNA remarkable as an information system isn't just its size, it's its density. 1 gram of DNA can theoretically store up to 215 petabytes of data. That's roughly around 215 million gigabytes. Researchers have already demonstrated this by storing entire films, books, and music collections inside DNA and retrieving them accurately. Imagine it. Everything on the entire global internet, every image, video, message, and document ever created, could theoretically fit inside a container that's smaller than your hand. And unlike hard drives, DNA doesn't need power to hold its information. Under the right conditions, like cool, dry, and dark environments, it holds for hundreds of thousands of years. We've read mammoth DNA. We've read ancient human remains. The molecule remembers what digital storage cannot. This isn't new technology we invented. It's the technology that invented us. The library doesn't just store information. It keeps receipts. Approximately 5,000 years ago, something dramatic happened to the human Y-chromosome. Genetic researchers reading DNA from populations around the world noticed that the male lineage diversity collapsed, sharply, suddenly, and globally. Where there had once been many Y-chromosome lines, only a handful survived. The best current theory is that it was coordinated clan warfare, where competing male lineages were eliminated and their women were absorbed into the societies of the victors. The X chromosome, passed maternally, showed no such collapse. That means the women survived. The men and boys of the losing line simply stopped appearing in the record. History didn't write that story down. The DNA did. Reading further back, the library reveals something else. When modern humans left Africa and spread across the globe, they encountered other humans of older branches, different lines. They interbred. Now, most people living outside of sub-Saharan Africa today carry between 1 and 4% Neanderthal DNA. Some populations across Asia and the Pacific carry traces of the Denisovan genome, a group so recently discovered we named them after the cave where their finger bone was found. These aren't just ancestry footnotes. Some of those inherited sequences shaped immune systems, altered metabolism, and changed how certain bodies respond to high altitude. The encounters written into the genome are still running today. The library holds surprises that previously taught history never anticipated. Genetic signatures found in certain Amazonian indigenous populations show unexpected similarities to populations related to Aboriginal Australians, a connection that predates any previously recorded contact by centuries. Someone crossed that ocean, and the DNA remembers the crossing, even when no other record survived. Every encounter, every migration, every war, and every reunion, the library kept all of it. For most of history, we could only listen to what DNA was saying. Now, we're learning how to write back. In 1994, a computer scientist named Leonard Adleman dissolved strands of synthetic DNA into a test tube and used them to solve a mathematical problem that would have taken a conventional computer much longer to figure out. He wasn't using DNA to study biology. He was using it as a computer. The molecule that stores life's instructions turned out to be capable of running calculations and doing so in massive parallel, processing millions of possibilities simultaneously in a space smaller than a drop of water. Since then, researchers have stored entire books, films, and music collections inside DNA and retrieved them without error. Microsoft has demonstrated fully automated systems that can write data into DNA sequences and read it back to you on demand. A company called Catalog encoded the entire contents of Wikipedia into DNA strands. The information held, and the molecule didn't care how long it waited. What makes DNA storage different from everything we've built before isn't just density or longevity. It's that the storage medium is alive in origin. It's shaped by billions of years of pressure to hold information accurately and pass it forward. Every error correction mechanism evolution built into the genome is, in a sense, free engineering. We didn't invent reliable data storage. We found it, and we're only beginning to understand what it can do. The library doesn't just remember the past, it's learning to carry the future. Reading the library changed what we now know. Writing to it is changing what's now possible. The tool that made this possible came from an unlikely source: bacteria. Among the oldest living organisms on Earth, developed a system to defend themselves against viruses. When a virus attacked, they would cut out a piece of the virus's DNA, store a copy of it in their own genome, and use it to recognize and destroy the threat if it ever returned. Scientists studying this immune system realized something extraordinary: that the molecular scissors bacteria used could be reprogrammed. You can point them at any sequence in any genome, and they would cut precisely there. That system is called CRISPR. In 2020, the scientists who developed it into a usable tool were awarded the Nobel Prize in Chemistry. CRISPR opened the door, but what followed refined it further. Base editing allows researchers to change a single letter in the genetic code without cutting the double helix at all. Instead, swapping one base for another with a precision the original tool couldn't achieve. Prime editing goes further still, functioning less like scissors and more like a word processor, by finding a specific sequence and replacing it with something new. In 2023, the first CRISPR-based therapy received approval as medicine. Casgevy, a treatment used for sickle cell disease, moved the technology from laboratory to clinic. For people whose blood cells had carried a painful and life-shortening era in their DNA since birth, one treatment rewrote the relevant sequence. The error that was inherited across generations was finally corrected. The library, for the first time, could be edited. But editing carries questions that science alone cannot answer. A treatment applied to a living person affects only that person. An edit made to an embryo, on the other hand, passes forward to their children and to their children's children. The line between correcting a disease and redesigning a life is not always obvious. But who decides what the library should say? Who has access to that pen? These are not distant questions. They are already here. We began this episode in a single cell. 3 billion base pairs, the equivalent of 200 volumes of text, 37 trillion copies of the same library that's distributed through every cell of every body alive on this planet. A molecule that outlasts empires, crosses oceans, remembers wars that left no other record, and carries traces of every encounter our ancestors ever had, including ones with humans whose names we never knew. We've spent most of human history not knowing this library existed. We've spent the last few decades learning how to read it, and now, for the first time, we're learning how to write. The code has been running for billions of years before the first human thought to look for it. It held the story of where we came from, who we met, what we survived, and what we passed forward, all of it quietly, in a molecule that's too small to see. Now we can see it. Now we can read it. Now, carefully, imperfectly, and with enormous consequence, we can change it. Every cell in your body is carrying a conversation that started before recorded history began. Now that we can hear it, read it, and for the first time rewrite it, the question isn't what the library says. The question is what we'll choose to add to it.
Cosmos, bring the patterns forth.
The whole concept of using biological systems as computational platforms is mind-blowing. The editing applications are where the real-world impact happens. The processing side is wild, though, using bacteria as living computers. So bacteria basically act like tiny biological processors. They use their natural cellular machinery to manipulate DNA sequences through enzymatic reactions. Kind of like how your computer's CPU processes binary code, but instead they're working with genetic information. The bacteria can read DNA inputs, perform logical operations on them, and then produce modified DNA outputs. Pretty wild that these microscopic organisms can essentially function as living computers. Actually, bacteria do use frequency-based mechanisms in their cellular processes. They communicate through chemical signals that create oscillating patterns, and their metabolic processes generate electromagnetic fields at specific frequencies. Plus, their DNA replication and protein synthesis happen in rhythmic cycles. It's like they have their own biological frequencies running their operations. While DNA computing in humans doesn't use frequency in the same way mycelium networks do, it does leverage the natural properties of DNA for parallel processing. DNA computing in humans relies on molecular interactions and enzymatic activities rather than electrical signals. Humans process DNA at the molecular level through transcription, where DNA is copied into RNA by RNA polymerase, then translated into proteins in the cytoplasm. This follows the central dogma. DNA encodes RNA, which encodes protein. Fascinating stuff. DNA processing moves forward through a series of precise chemical reactions. Enzymes like helicase unzip the DNA double helix, while DNA polymerase adds new nucleotides to build the complementary strand. It's like a tiny molecular assembly line, with each enzyme playing a specific role in the replication process. Chemical reactions do involve frequencies, but not in the way you might think. When molecules collide, they vibrate at specific frequencies. These vibrations can affect reaction rates. It's like a molecular dance, where the right moves at the right tempo lead to a successful reaction. Human DNA computing does involve frequencies, but in a different way than mycelium networks. The molecular vibrations during DNA's chemical reactions create specific frequencies. These vibrations affect how enzymes interact with DNA, influencing the computing process at a molecular level. The key difference is in how they use frequencies. Mycelium networks actively use electrical signals and low-frequency sinusoidal waves for communication and computation. Human DNA computing, on the other hand, involves molecular vibrations as a byproduct of chemical reactions, not as a primary communication method. DNA is basically our living history book. The Y-chromosome bottleneck around 7,000 years ago shows how warfare and social changes literally rewrote our genetic story. It's wild that we can read these ancient events in our genes today. Aboriginal Australians made it to South America thousands of years before any European explorer. The genetic evidence completely rewrote what we thought we knew about ancient human migration patterns. It's wild that we've got this incredible power to rewrite our biological code, but we're still figuring out the governance part. Right now, it's mostly scientists and regulatory bodies making those calls. This affects all of humanity. You're absolutely right to be concerned. The sickle cell treatments are amazing breakthroughs, but there's definitely a fine line. International regulations for DNA editing are evolving. The US and UK use a product-based approach, focusing on the final organism, while the EU is shifting to allow new genomic techniques for crops. There's a global consensus against heritable human germline editing, but no binding international treaty exists. Fascinating pattern here. You started with DNA as a biological processor, moved through its role as a historical record keeper, then landed on the ethics of who controls the editing pen. That's basically the past, present, and future of genetic power in one narrative. The pattern I see is how DNA keeps shifting from being something that happens to us to something we actively control. And your episode captures that exact transition point where humanity becomes the author of its own genetic story.
If this kind of exploration resonates with you, you're welcome to join me here by pressing like or subscribe.